Hey everybody, welcome back. So now we're going to talk about microbial growth and how the growth occurs in nature, and then we'll talk about laboratory conditions. So we study microbes in general in laboratories, and we have control over how well they grow when they're in a laboratory condition. But in nature, the conditions that microbes are growing in change a lot. And this affects microbial growth and microbial behavior. And so the cells have to be able to adjust to the changing environment. And they do this by sending out signals to determine how many cells are in the environment. They can sense different chemicals, the changes in those chemicals, like chemical changes, and they can produce responses appropriate. If they can't do this, they'll die, and other microbes will come into the environment. So in general, when microbes are in nature, they grow in communities. So they can live singly, but most grow in large polysaccharide communities termed biofilms. And so this is an example of a biofilm. Biofilms are found all over in our natural environment, including in our own homes. So you can find biofilms on stream beds. So if you ever felt the rocks along a pond and they're kind of slippery and mucky, that slimy muck, that is the biofilm. They're found in sink drains, so the gunk in sink drains, the scum in the toilet bowl, the muck on the edge of your tub, on the around the tub oftentimes can also grow. You can get scum on tubs, and you can get biofilm formation on your teeth in the form of dental plaque. So plaque is a form of biofilm. Biofilms are very, very protective of their cells. They start out with individual cells adhering to a surface. That surface typically is nice and clean. The cells adhere, they start to replicate, and they start to produce polymers. These polymers are what cause the slimy appearance on biofilms or of biofilms. And the polymers also allow other cells to come and attach. Once more cells start to attach, the bacteria are now no longer able to free themselves from that original surface. And so they're going to gain nutrients from other bacteria. So the nutrients that are used by some bacteria are going to be utilized, and then when they produce waste, those waste products can be used by other bacteria as nutrients. And the cells can communicate with each other as well in those biofilms. The only cells that are typically prone to any damage are the ones on the edge of the biofilms. So biofilms are really important. Medically, most infections involve biofilm formation. And biofilms are resistant to antibiotics and our immune system. So there's this biofilm here, here's the antibiotics. Bacteria are like, ha ha, funny, funny, right? Because they're in the biofilm. And there's some viruses and other microbes trying to get into the biofilm before the antibiotics can attack them. Like I said, the only cells that are actually at risk are the ones on the edge of the biofilm. Other than that, the other cells are protected. The only effective mechanism of removing of a biofilm is mechanical removal, physical scraping of the biofilm, which is why every single morning and every single evening, you are told to brush your teeth. That helps to get rid of the biofilm that is starting to form. It can also be a problem in industry. You can get biofilm formation that causes pipes to not be able to be used properly. Drains can be plugged just because of the biofilm. And then biofilms can also be used beneficially in wastewater treatment. We use biofilms to help to filter our water more effectively. So there's examples of biofilms in the environment. And in biofilms, in microbial communities, the interactions between cells is oftentimes called cooperative. A cooperative interaction occurs when microbes work together. So you might have anaerobic cells as well as aerobic cells in the same environment. The anaerobes can only grow outside of oxygen. They can't grow in the presence of oxygen. So if we have aerobes taking all the oxygen, then anaerobes are able to grow in that aerobic environment because the aerobic organisms utilize all the oxygen. And as I said before, a waste product from one cell can serve as a nutrient for another cell. But sometimes interactions can also be competitive. And so in this mechanism, cells will produce toxic compounds that inhibit other cells from growing, and this allows those cells, the compound producer, to survive and replicate more effectively. An example is gram-negative bacteria oftentimes use a structure known as a type 6 secretion system to inject toxic compounds into competing bacteria. So those cells die, and the gram-negative cells can replicate and take over the environment. I'm going to stop here, and we'll get into growth in laboratory conditions in our next video. All right, talk to you soon. Bye.